Hello, I'm Dr. Kaustav Majumdar. I'm an associate professor in the Department of Food Science and Technology at the University of Nebraska-Lincoln. Today, I'm going to talk about food proteins, peptides, and the connection or the relationship on human health. So that is my primary research area in UNL. I works with different kind of dietary proteins and peptides and their effect on human health. So when we start talking about proteins, we first need to think about this, that protein-rich foods are one of the earliest food for mankind. We invented fire, we invented the weapons, how to hunt, and then we start hunting and cooking the food with the fire. Potentially, protein-rich foods are, I think, the one of the earliest food that we cooked and eat. So talking about proteins, it's a very diverse biomolecules. It's different shape, size, and is there. For example, for hemoglobin, like we all know hemoglobin, what's its role in our blood. Like we have these hemoglobin, which is a monomer for some species, and some species it is like 144 more SN. So it's a similar function, but they look very different in different species. Now, if we talk about the basic chemistry of the protein, we go back to the amino acid, the building block of a protein, which have a carboxylic end, the acid end, or a basic end, or an alkaline end, which allows them to join with each other and form polymers. So you have this amino end and this carboxylic end, and it's the only form of source of digestible nitrogen for human. Now, these peptides come together and form a peptide bond. Like this is the uh, two side. This is one amino acid. This is another amino acid. The carboxylic and the amino end in another amino acid is come together and form a peptide bond. So if it's two amino acids come together, that's a dipeptide. If the three amino acids come together, that's a tripeptide. And if it's more than that, we can go say it's a polypeptide, right? So one end of the amino terminal and other end of the carboxy terminal. So in a polypeptide chain, one end should be a amino terminal, the other end is a carboxy terminal. So this is basically a condensation reaction when this carboxylic group and amino group come together and form this peptide bond. Now, this is a regular peptide bond. There are other different unique kinds of peptide bonds are there. So one of them is this isopeptide bond. So this isopeptide bonds form when you have the amide bond between the side chain amines or carbonyl carbons on the side chain on the other side rather than the alpha carbon. So for example, the glutamic acid up here. So this is the alpha carbon. This is the carboxylic end, the regular carboxylic end. And then, but if you go up here, there is another carbon up here, which is a gamma carbon, but there's also a carboxylic end up here. So if a peptide bond that is formed using this carboxylic group, not this one, so this is a regular bond. This is a gamma bond, so it will be calling it gamma peptides. For example, glutathione. You see the gamma glutamate using the carboxylic group on the gamma carbon and then forming the peptide bond with the cysteine up here, and then the cysteine binding with the glycine with the regular peptide bond. So this, because of this bond, it's a gamma peptide. You also have a beta peptide. So it can, instead of the gamma one is binding with the beta one, for example, the beta alanine L-histidine, which is a carnosine, or beta alanine 1-methyl L-histidine, which is an arnisine. So these are the beta amino acids. So technically, these are the beta peptides. So these both alpha peptides and beta peptides, they are unique because they are relatively a little bit rigid, on protease or digestion or proteolytic because proteolysis because of this unique bond. So that's why they are unique peptide bonds. We also talk about the other unique bonds, which is a circular peptide. So here at amino terminal and the carboxy terminal, they come together and form a bond. So it's become a circle. So that's a cyclic peptides. So these are everything is still an alpha peptide or regular peptide bond, but they arranged as a macrocycle. So, for example, the bacteriocin or cyclosporine, the plant cyclotide, these are all circular peptides or cyclic peptides. So it's have the different amino acids, they come together and then form a circle. Now, we talked about the bond formation. There is also the hydrolysis of the peptide bond, which is a break down the two amino acids here. So this is a peptide, this is a peptide bond. Now it's break down to two different amino acids, like the form amino acid of this one. So this is a basic protein breakdown. This is a hydrolysis process, which can be induced by different things like pH, heat, or enzymes, proteases enzyme. For example, let's have the acid hydrolysis, which is a non-specific peptide cleavage. It's go and just bind, so break down as the acidity go up. You can also happen with the enzymatic hydrolysis, where it's just action of a specific enzymes like proteases. We have the enzymes like pepsin, trypsin, chymotrypsin in our GI tract. We also have other proteases, like bacteria have proteases like alkylase, or plant protein like the papaya have papain, or pineapple have bromelain. These are different kind of proteases that exist. Now, talking about enzymatic digestion or hydrolysis or proteolysis of proteins to formation of peptides. So this is a typical protein structure. It's a schematic. So this is an amino terminal and the carboxy terminal. It is a different amino acids that they bind together with these peptide bonds. So the red dots are the amino acids and the blue lines are the peptide bonds. Let's think it that way. Now, when we see an endopeptide is this, if it's cut, so endopeptide is a digest the internal peptides bond. Also, this one, like this, it will digest the internal peptide bond. Whereas the exopeptidases digest the peptides bonds from the terminus. So it will do the break the bond up here, break the bond up here. So now after that, they will produce peptides or they will produce free amino acids. Sorry, not or, peptides and free amino acids. So you will have small peptides or smaller peptides, whatever it was exactly how much big it was, it will be a smaller ones, and then we have a free amino acids. So talk about the gastro-intestinalization of food proteins. Like we eat a dietary protein, then it goes break down through pepsin to digested protein, and then this digested protein goes down to small intestine. It's broken down by the pancreatic enzymes. You have undigested proteins, you have a peptides and amino acid that got absorbed. The other ones go down to the colonic formation and they're fermented by the microbes in our colon, and then we produce multiple other metabolites or short chain fatty acids or different kind of other peptides, the rest going out of the feces. This is a typical way our protein, the dietary protein get digested in the GI tract. So you have the enzymes that are present, are all are endopeptidases. These are pepsin, trypsin, and chymotrypsin. There are some exopeptidases also there, which is the aminopeptidases and carboxypeptidases, which primarily in the brushwater membranes in the small intestine. Now, once you digest these proteins into peptides and amino acids, this amino acid is going to absorb. So amino acid goes from the lumen of the intestine to the bloodstream, specifically to the portal vein and then to the liver through the amino acid transporter. So we have this amino acid transporter in the gastrointestinal epithelial cells, which takes these amino acids from the lumen and then transport into the bloodstream. But these are different kinds of amino acid transporters are there. For neutral amino acids, we have the different kind of transporters. For diabasic amino acid, you have the dicarboxylic amino acids. We have different transporters. So there is different ways these transporters are present there. And if you do not have these transporters work properly, you have all of these diseases that I listed up here. So now, from amino acid to peptide. Now, amino acid is one single amino acid is going through all these transport system that is listed up here. But when it's a peptide, which is two amino acids bind together, at least two amino acids bind together, form a peptide bond, and to absorb the peptide, it's going to absorb in three different ways. It can be absorbed by a transporter called PepT1, which is a peptide-specific transporter. It can move through the transcellular movement with peptide as a cargo, or it's a paracellular movement. So it can go through a specific receptor and then go inside the cells, and then it could be broken down here, or it can remain intact and then go to the bloodstream. Here, it can penetrate the bilayer, go through the endocytosis process, and then got absorbed, or it can be absorbed between the paracellular route, between the gap between two epithelial cells, and the peptide got absorbed into the bloodstream. So this is the way the peptide got absorbed. Now, there are different factors that affect the peptide absorption. So, for example, for uh, to efficiently absorb the peptide, you need a proton gradient that is created by the hydrogen. And then flavonoids and fatty acids helps in this proton gradient, establishing the proton gradients. Sometimes there are competitive inhibitors that are present. Some peptides are competitive inhibitors of peptide-1, so which actually inhibits the transport. Also, sometimes the translation regulation, like amino acid and other protein hydrolysates, or different kind of aging process, for example, one of them, or different kind of disease like cardiovascular disease, primarily hypertension. This affects the transcription and regulation of peptide-1, so it affects your uh, absorption. Also, the theaflavins, the phytochemical, that's actually downregulate the peptide absorption. Now, in vitro system, we can analyze this peptide absorption through a transfer membrane system, where you have this layer of filter where you grow cells, even gastrointestinal epithelial cells on the top, add your peptide up here, take your samples from the bottom layer, which you call the basal layer or basolateral layer, and then you analyze through LC-MS to find out what are the peptides that got absorbed up here. You can also use this instrument called Using chamber. It's basically do the same things up here, but you can use the gastrointestinal epithelial cells of a mice, or just the whole GI tract, not the whole, at the portion of the GI tract that you can put it up here, and you can simulate the environment of the gut, and then can absorb the proteins, amino acids, and peptides to that. Now, you can also detect how the peptide is going to absorb in vitro. In order to do that, we mostly do these animal studies, the rat or mice studies. Rats are specifically better because we generally collect these samples from this portal vein. So portal vein is the vein which takes all the nutrients that got absorbed in the stomach, in the small intestine, and take it to the liver. So the portal vein, if you want to take it from here, if you want to take the blood from here, rat is a better model to do that. But this portal vein then take all of these peptides, go to the liver, and then liver throws down to the hepatic circulation. After the hepatic circulation, throws down to the whole body. And you can use different kind of mass spectrometry to detect these absorption of these peptides. Now, we initially think this is one of the biggest myths, that whatever we eat, all our dietary proteins got broken down in our GI tract into amino acid, and we only absorb amino acids, which is not true. We have specific mechanism to absorb di or tripeptides, some other peptides like quadrupeptides or pentapeptides, 4 or 5 or 6 amino acids long peptides can get absorbed through transcellular pathway, which I showed before, or the paracellular pathway. So there are different ways that our body absorbs peptides. And we have also shown, like the scientists, people have also showed that the peptide can actually cross blood-brain barriers. So dietary peptides can cross blood-brain barriers. So not all of them, but some of them, the peptides which maybe potent one, can actually cross blood-brain barrier. This is a study that came out in 2019 in scientific reports from Dr. Toshiro Matsui labs in Kyushu University from Japan. And they used one peptide called tyrosine protein, which transported across the blood-brain barrier with the concentration of 3.53 microliter per gram. So that's specifically the Ki value of those peptides. So intact peptide can get absorbed by the human gastrointestinal system, and if it's then stable, then this peptide can cross blood-brain barrier and can show different kind of biological effects. Now, talking about the effects of peptides on human health. So peptides are ubiquitous biomolecules, and the language of our body. That means like that when two cells of our body talk with each other, they communicate with each other, they use the signals, right? And these signals are primarily peptide-like structures. We also call them hormones, but they are essentially peptides. For example, the defensins or the bacteriocins or mucin and the very famous insulin. So these all are peptides, and these all have different biological activities. So peptides essentially have a specific role. Each peptide have a specific role in our human body. So that's why these peptides are biologically active. Now, what is bioactive peptides? Like what is specifically food-derived dietary bioactive peptides? So these peptides are generally inert in their native protein sequence. That means that if this is the parent protein, this is not this specific section, this specific sequence may be bioactive, but they will be not showing any activity until unless it's get out of this protein and then go out, then you see the biological effect. Now, these peptides can produce through food processing, can produce during gastrointestinal digestion, can be naturally occurring in foods, or also we can produce them through chemical synthesis. But the major challenge of these bioactive peptides or BAPs are their stability in the GI system. So if it's produced in the food processing or it's present naturally in the foods, it will be broken down into maybe small amino acids or small other fragments of peptides that it will not get absorbed in the bloodstream. The other point is that sometimes when you have these peptides produced, suppose it is produced through a food processing techniques like enzymatic hydrolysis or things like that, it will give a bitter taste. So that's the biggest challenge of these peptides. So they are biologically active. They can mimic the function, the different bodily function, or they can control different bodily function or regulate different bodily function. However, you can release it from food proteins, but they can be very bitter as well. So there are different ways that peptide discovery happens all over last 20, 30, 30 years or almost. Specifically, people generally choose a specific food protein, just choose an enzyme, hydrolyze them, do multiple chromatography, fractionate them, measure their biological activity, select a protein fraction, do further chromatography, and then identify one or two sequences that are biologically active. This is a long and very cumbersome process, very labor-intensive process. That's why people start developing different approaches that uh, could make it more efficient. So what people never justify, like which protein to start with, like which food protein we should first look for these bioactive peptides, and what enzyme you want to use. You want to produce it externally. So that's how we need to develop different process. And for that, the modern approach that came out is here that we know the protein sequences. No, if not all, but majority of food protein sequences are present in the public database. And with the advanced computational chemistry, we can now specifically select, or we can simulate the action of an enzyme. And if we can simulate the action of an enzyme and then use that that protein sequence and this and then we can get a theoretical peptides that could be produced from an action of a particular enzyme. And once you know which peptides can be produced from this protein, then you can use something called QSAR, which is a quantitative structure activity relationship, and predict the activity of the peptides. Now, with the recent advancement of machine learning and other computational chemistry tools, development of, say, dynamic molecular dockings and things like that, these cumbersome process or lengthy process can be achieved very fast, and we can be more efficient of identifying and evaluating the value of this biological activity of these peptides. Now, This is the typical way I was talking about, is go for protein sources, do protein composition analysis. You can do in silico enzymatic hydrolysis, produce these peptides. You can use the same combination using in a wet lab, measure the activity assay, use a mass spectrometry, and then identify the match the two things and find out which are the identify the bioactive peptides, which are the ones which potentially screen for the biological activity, and you can go further. And then through that method, we can develop further with dynamic molecular dockings and things like that, use more of a computational chemistry to identify these peptides. Now, if you look into last 30 or 40 years of research on bioactive peptides, the majority of the research, which is the bigger portion of the pie, are ACE inhibitory peptides. Now, what is ACE? ACE is angiotensin converting enzyme. So this angiotensin converting enzyme is an enzyme that's is a key regulator of our blood pressure regulation. So the majority of the peptides research that actually focused on ACE inhibitory peptides. There are also antioxidant peptides, antimicrobial peptides, DPP4 inhibitory peptides that can help in regulating your blood glucose level, the cell toxic peptides, the peptides which have a cell toxicity effect, the opioid active peptides, the neuropeptides. These are all different kinds of peptides, anti-inflammatory, antithrombotic, and then you can see the membrane active peptide, you can see the opioid agonist peptide, you can have a vasoconstrictor peptide. So there are different kind of activity people have identified so far. But in the interest of time, I'm going to focus today on few biological active peptides that people have done most research on so far. And the biggest one, as I said, is the angiotensin converting enzyme, or ACE inhibitory peptides. ACE inhibitory peptides. In this case, what happens that ACE is that enzyme that actually produce in our lungs, which converts angiotensin 1 to angiotensin 2. Now, angiotensin 2 that actually increased our blood pressure. Now, if we could inhibit the action of angiotensin converting enzyme ACE, you can control our blood pressure. And the first blood pressure medication, or ACE inhibitory medication that actually isolated from the Brazilian pit viper. So the British pharmacologist Sir John Vane studied this venom on the Brazilian pit viper, and then developed the first ACE inhibitory to control the high blood pressure. Now, in terms of dietary bioactive peptides, the two very well-studied peptides are these two peptides, IPP, valine proline, and isoleucine proline. These are all coming from milk. So this is a product that is you can find in Japan, which is Calpi sour milk, which is essentially a fermented drink, which uses Lactobacillus helmictus, and originally it was used Saccharomyces cerevisiae, but right now they use Lactobacillus casei, DeBruckii, and ramosus. This combination of these three bacteria, and then produce this, after this fermentation, they produce this sour milk, which have these peptides, the VPP and IPP. And this can reduce your blood pressure, like in this is the data coming out from a rat study that reduce the systolic blood pressure effectively in these rat studies. Now, this is a pretty old study, like almost like it's actually 30 years ago. Now, from since then, we start understand more about these two peptides, this lactopeptides, VPP and IPP. These lactopeptides can actually control your arachidonic acid system. It can control your vascular function. It can control the blood coagulation system. So it's have multiple effects in modulating heart health. So it's pretty much not only the ACE inhibitory, that's the central point up here, but along with the ACE inhibitory, it can increase the COX-1 expression. It can do the eNOS expression. It can increase the eNOS expression. It can reduce ICAM1 and VCAM1 expression. It can reduce NF-kappa B protein. It can decrease MCP1. So these are all the biological effects, mostly focusing on heart health. These two peptides, IPP and VPP, can do. Now, people have done different kind of human clinical studies. as it was first identified in Japan. That in Japan there was a lot of different kind of studies that have been done, like human clinical trial that have been done with IPP and BPP. Personally, when I was in Japan, I actually tasted this Calpis sour milk. It's pretty nice to drink that. It's a bit sour, but it's pretty nice to drink. And their through their Japanese foshu system, they actually have a way uh, uh, the, to actually claim the health beneficial biological effect of particular food products, and that's why they did uh, all of these studies with these two peptide IPP and VPP, the human clinical trials. Now, these human clinical trials, if you see that, they primarily consume 5 milligrams of these peptides per day, which had reduced the blood pressure roughly around 6 millimeter of mercury. So that's the kind of, if you summarize all the meta-analysis for these two peptides. Now, when these peptides were trialed on European population, in the Dutch population, you can see the IPP-rich milk protein hydrolysates. It's not a fermented milk, it's a hydrolysate, which reduced the BP by 3.8 millimeter of mercury. So it's a little bit less effect than what's showing, what people have observed in the Japanese study. Then there is one study that I'm going to talk about here is that's actually, I think this is the Finnish study, and then which actually did all these meta-analysis again for the IPP and BPP that people have done on specifically on a human clinical trial. You can see the peptides can effectively reduce the blood pressure in a moderate reduction of 1.5 millimeter of mercury in the European subjects, where in the Asian subjects it's a little bit predominant, more effect on that. And that's because of maybe the epigenetic effect, the absorption of the peptides, the absorption efficacy of these peptides into different population, the effects of other dietary factors. People talk about the effect of gut microbiome is different, and other different kind of foods that they eat. The Japanese population primarily is eating, or the European population is primarily eating. That's maybe hindering the absorption or the biological activity of this peptide. Now, from acinetylpeptide peptide, I'm shifting the gears to lactoferrin, which is an antimicrobial peptide. So, which is coming out from human milk, first identified from the human lactoferrin, which is a particular peptide that is have the most antimicrobial activity, which is lactoferricin. So, this is the structure of lactoferrin. And you can see this yellow portion up here, which is lactoferricin, which is typically corresponding to 1 to 47 terminal region of this particular protein. And lactoferricin consists of like 18 loops that forms by the disulfide bridges. So it's a pretty big peptide, which have 18 disulfide bridges up here. Remember, the previous two acinetylpeptide peptides are tripeptides, VPP and IPP, only 3 amino acids, right? But this one is much bigger and have this much complex structure. So this lactoferricin that show antimicrobial activity, they actually, this is the mechanism of action of these particular peptides. This is a positively charged peptide, just bind with the negatively charged outer membrane, and then the cationic side bind with the LPS and interact with the hydrophobic parts of the membrane, and they interact with the hydrophobic heads of the membrane, and then aggregates, form the mycelial complex, do a flip-flop across the membrane, and then interact with negatively charged cytoplasmic compounds like DNA and RNA, and then do the autolysis of that. That's how it's actually exhibit the antimicrobial activity. There we talk about the antioxidant peptides. So these are the peptides which can reduce the reactive oxygen species level in your body, reduce oxidative stress. And this is primarily two ways it can work. It can work as a radical scavenger, which peptide that scavenge the free radicals, suppose the superoxide. It can also act as an enzyme inhibitor. For this is a class of peptide that can inhibit the action of an enzyme responsible for the free radical production, like NADPH oxidase inhibitor. And it can also have other enzymes that can enhance the expression of, say, catalase, which can break down H2O to water and oxygen. So these are the three different mechanisms. It could be a hydrogen atom transfer mechanism, single electron transfer mechanism, and solvent-assisted proton loss. So these are the three different mechanisms where antioxidant peptides exhibit their biological effect. There is one more up there, which is a metal chelating effect. So it's bind with a specific metal. Here I'm showing as iron with a histidine residue, and then it can reduce the antioxidant, express the antioxidant activity. This is the last thing I'm going to talk about is the bioactive peptides, which is for topical application. So the three previous application I'm talking about was acidic peptides, the antimicrobial peptides, and then we talked about the antioxidant peptides. But this one, so all of those you can act inside the body. So you need to eat the peptides and go in and then can show the biological effect. So for this one, this is collagen peptides, which can have a topical effect. So you can use on the top of your skin. For example, this is you're showing an wound healing effect. So this is a vehicle which did not really heal properly, or where you have this high dose of these peptides, it's healed much better. The low dose also healed pretty good. So this is the one effect of the collagen peptides. So you can probably see these collagen peptides and the vital proteins things in your Costco or your other nearest supermarkets. So this is pretty popular these days about this topical use of these peptides. The last parts of it, that these peptides, as I previously mentioned, can also exhibit elicit taste. We talked about the bitter peptides in the beginning, but not only bitter, it can actually also do umami. It can also do sweet. So these are different kind of peptides and can produce the same kind of sensation and can have different. So we can actually modulate the taste of a compound through the addition of peptides. So that's primarily what my lab does. So my labs primarily look at the gamma glutamate peptide. So it's also an isopeptide, which is cuckoo active. So once these peptides can actually bind through a receptor called calcium sensing receptor and can reduce vascular inflammation, we have shown we have demonstrated that with these peptides, we can reduce the atherosclerotic plaque development in mice. And these peptides are you can find in different kind of pulses, so you can sprout the pulses, but these peptides can go, the abundance goes up a little bit. And we are also using other processing techniques like fermentation, as the VPP and IPP. Remember that we used the fermentation for milk protein. So we are using the similar fermentation techniques, but using pulse protein to produce all of these peptides, which these cuckoo active peptides that can have biological health benefits in mice. So at the end, I would like to finish this talk with this thought. So if you have one amino acid, you have 20 possible variations. That's a 20 amino acids. When you have dipeptides, you can have a maximum of 400. When you have a 5 or pentapeptides, this is the number. When you have a 10 or 10 amino acid together, so this is the number. 30 and 500, that's the number. So the diversity, the different kinds of peptides that's present, it's pretty large. And typically bioactive peptides are from 2 to 10 amino acid long. So this, if you add all these numbers, that's different combinations of peptides that can be possible. And from that, some of them will be bioactive, some of them will be not bioactive. But our job is to identify which of these sequences are biologically active. So with that thought, I would love to meet you guys next week. And if you have any specific question, we'll try to answer that. So thank you so much for listening to it. And I would love to talk when we meet in person next week.